Hey everyone, and welcome back to episode 3 of What If by So Cultured. We just want to take a quick moment to say that if you guys are enjoying this season so far, or just our podcast in general, please take a moment to give us a review or a rating on Spotify, Apple Podcast, Amazon Music, whatever it is that you are listening to us on. We would really, really appreciate it. But before we get started with today's episode, we are, of course, kicking off this intro with another deep philosophical question because Taz posed this question to me right before we started filming, and I have some thoughts. So Taz, can you tell the listeners what you asked me before we started filming? Because I was like, no, you need to save this for the show because the listeners need in on this conversation.
Okay, so my question to Liv, because basically, if you don't know, I'm always based in Australia. Liv is always based in the UK. And so we're always filming like at completely opposite times. Usually it'll be my breakfast time and Liv's dinner time, or vice versa. Obviously today it's my dinner time and Liv's breakfast. So my question to Liv was, why is it breakfast the only meal with rules? And basically what I mean by that is, I feel like with lunch and dinner you can literally eat and drink anything, but with breakfast there's like a set of kind of unwritten rules, like unless you're at an airport you're not having a pint before 11 a.m. I'd say. Like,
I think that's a British
a given
thing. Like, I
Maybe.
think I genuinely think that is a British thing. Like, we say time doesn't exist when you're in the airport. You are allowed a pint of beer at 8 a.m, 6 a.m. if you want. If you're in the airport, there's no rules. Aperol, 6 a.m.
Well, yeah, exactly. That's so alcohol being one, that would just be really weird. And then another one is like pizza for breakfast. I don't know, like a burger for breakfast. But then you could have like breakfast foods for dinner. Like, I've definitely eaten a bowl of cereal and like some toast for dinner before because I can't bother to cook. So that is my philosophical question of the day to you.
I mean, yeah, when I was like at uni, or just, I think genuinely like from 19 to 23, I don't think I made dinner like ever. Like, I would literally just have cereal or toast for dinner every single night because I'd always get in. Yeah, I'd get in so late and I'd be like, oh, fuck it. I just can't be asked. I'm going to make some toast. So unhealthy. But you are right that there is, but why? I mean, the alcohol one I get, right? Because you don't want to start your day off on alcohol. Again, this might be like a cultural thing, and I think it is, but why am I not allowed chicken curry at 8 a.m. if I want it? But I don't want it though. Is that just ingrained in me? I don't want it.
Yeah, that's the thing. I think like heavy, rich foods in the morning is just a bit, I don't know,
too much.
Blasphemy, isn't it?
But when you go on like holiday and stuff, like there is the option to have curry for breakfast if you want. So it's not, I think it is a cultural thing. It's not like just humans in general don't want to eat curries. So at 8 a.m, do you know what I mean?
One thing I will say actually, and I don't want this to come off as offensive, but this was a culture shock for me. When I went to the Philippines, we were in like a really rural part of the Philippines, and they gave us what is considered, I guess in that region, a Filipino breakfast. And it was a bowl of rice with fish and garlic and I think an egg on top. And for me, that was way too much for breakfast. Like, I would eat that for dinner, don't get me wrong, but it was very early in the morning and I'm like, this is actually the last thing that I want right now. Like, give me some toast or something.
I know what you mean, but on the flip side, I also remember when I went to Florida and their breakfast is like sugar, 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 sugar. And I'm like, that Filipino breakfast is a better way to start your day, 100%.
Oh,
because
yeah,
when I went
way
to Florida,
healthier for sure.
Yeah, they've got stacks of like I'm talking stacks of pancakes like this, and it's like drenched in like golden syrup, sprinkles, chocolate, like all of this stuff. I was like, surely that is literally the worst way to start your day. America in general, their food is crazy. I'm not gonna lie, like the portion sizes, I'm like, oh no, I actually could not. I could never. It's insane.
Yeah, it is. But I guess like, yeah, we've not really answered our question. Like, why is breakfast the only meal with certain rules?
Each individual like country has their own rules when it comes to breakfast.
Oh, for sure.
I think.
Yeah.
Like America, you want a plate full of sugar, go for it. So I think each country has their own rules, and our rule in Britain is if you're in the airport, you can have a pint of beer at 6 a.m.
You
That
can.
is the most important rule. God, anyway,
knows?
we're just gonna be talking about breakfast for this whole episode. Guys, can you give us your thoughts on the breakfast rules? Do you have any personal breakfast rules that you have set for yourself? Because we want to know. But for now, let's get into today's episode. Welcome to the So Cultured podcast,
where we explore science and tech unfiltered.
We're your hosts, Liv and Taz, two girls who love STEM and spilling the tea.
Each season, we take on a new theme, whether it's busting conspiracy theories, celebrating women in STEM, or breaking down the science in your favourite sci-fi films.
And of course, in every episode, we spill the laboratory,
where we share your wildest stories from the world of science.
Let's get into this week's episode of So Cultured.
Imagine if you could turn back the clock, not just superficially, erasing wrinkles or restoring grey hair, but at the cellular level, rejuvenating the body itself. Imagine a world where you and your loved ones could live longer in good health, free from the chronic diseases that plague our senior years. For thousands of years, humans have dreamed of this, from myths of the fountain of youth to ancient stories like the Greek myth of Tithonus. The idea of slowing or reversing aging has long felt like fantasy, but today, science is beginning to make it tangible.
Oh my God, I love this topic so much. Do you know one of my first ever pop science, when you used to actually go into Waterstones and go through all the science books and you pick a book, it was just the most exciting thing ever. One of my first ever books was actually from a guy named David Sinclair, who I know is a bit controversial now, but back then everybody loved him. But he's literally written a book about aging, and it's so interesting to me. So I'm very, very excited for today's episode, and the listeners should be too. But first, let's talk about, because I know this is something that he spoke a lot about in his book, and it's really interesting. So can we delve into first why, what actually makes us age? Why do we age?
Yeah, so obviously we know aging is not just cosmetic. Aging is the underlying cause of the vast majority of diseases and death. And aging is basically caused by the gradual accumulation of molecular, cellular, and tissue damage, what some scientists describe as the ultimate wear and tear of life. So unlike cars or machines where parts can be replaced easily, our bodies accumulate damage at the microscopic level. And this is typically from things like oxygen radicals, from metabolism, radiation from the sun, and normal cellular processes that all cause tiny breaks and errors in our DNA, proteins, and cell structures. Now, our cells do have repair mechanisms. Stem cells replenish tissue, DNA repair enzymes fix mutations, and cellular cleanup systems remove waste. But over time, these systems become less effective. So senescent cells, which are basically cells that have stopped dividing but don't die, accumulate and release inflammatory molecules that harm the surrounding tissues. In addition to this, stem cell numbers decline, impairing tissue regeneration. DNA errors accumulate over time, and organ systems gradually lose their capacity to function optimally. And this is why the older we get, the more fragile we become, and the higher our risk for diseases like heart disease, diabetes, dementia, and cancer. And aging itself is actually the single biggest risk factor for most major illnesses. So what's really important here to think about is there's no single cause of death called old age. So people don't die of old age. People die when critical systems fail. So the heart, brain, the immune system, or other organs, and aging increases the likelihood of these failures. And it's a process that's shaped by both physics and biology, wear and tear at the molecular scale, coupled with declining repair mechanisms, basically.
That's so interesting when you put it like that. And you can see why, a lot of scientists have such a strong interest in figuring out why we actually age, and then also if you can actually stop aging. But I think it's really interesting from an evolutionary perspective, because I always think what is the purpose of it? It almost makes you think is there a deeper get a bit deep here, but like, is there a deeper meaning to it? Like, because why do our bodies not just like maintain themselves perfectly? Do you know what I mean? Like, why do we not have processes that like avoid all of these things happening? Do you know what I mean?
Yeah, no, 100%. And I also had the same thought, because when I was doing my research for this, the chances of a human dying increases kind of exponentially after the age of about 60. But there's other animals, like certain species of tortoise, that will just sit at like 1% chance of death until they're like 200 years old.
Yeah.
So I think it's really interesting to look at it from an evolutionary perspective, but the answer is nearly always reproduction. So there's this theory called the disposable soma theory that suggests that organisms invest energy in reproduction rather than indefinite maintenance. So in the wild, organisms are unlikely to survive long enough for the perfect repair mechanisms to matter. So evolution then favors early reproduction over lifelong preservation.
Yeah, I guess that does kind of make sense, like when you think about it, because didn't they used to say like we used to not live until like past the age of like 40 anyway?
Yeah, no, exactly. In a world of disease, living past 40 or 50 was actually rare. Obviously, today, life expectancy has risen dramatically due to scientific and technological advancements. So obviously, vaccines and antibiotics, and also basic hygiene, were very key players in this. So obviously, before that, our bodies didn't have the capability of, I don't know, getting rid of tuberculosis or polio, you know. So it's kind of these scientific advancements that have led to our life expectancy to become longer. But obviously, with longevity becomes a brand new challenge. So we actually now spend more years in poor health than ever before, because many people live decades with chronic pain, frailty, or cognitive decline. So medical researchers are shifting focus from merely just extending lifespan to actually optimizing health span, which is the period of life spent free from disease and frailty. And foremost, the goal is not necessarily to live forever, but to live healthier, longer lives. And the evidence suggests that this is achievable. So you've got centenarians, which are people who live to be over 100. They're the best evidence for this, because they often remain independent and active and healthy until very shortly before death. So they're very rarely suffering for long periods of time. So that demonstrates that biological aging can, in principle, be compressed or slowed as well.
Yeah, this is really interesting. There's like a whole like epigenetics field like looking into this, and my old supervisor at Essex, he was like really interested in like aging, and like he would always talk about it, and I just thought it was the most interesting thing ever. But my life goal is to be a centenarian.
Yeah, same.
I want to be a centenarian. I'm convinced that my grandma is going to be a centenarian, because she's like in her 80s, but if you met her, you would think she's like in her 60s. She's so active. Like she's so like biologically young. So if she's not a centenarian, then there's no hope. And I'm related to her, so maybe I could also be a centenarian. It's my goal.
I feel like that's actually been a goal of mine. Like, remember people used to turn 100, then they'd get a letter from the Queen. Like, I still remember that as a kid, and I'd be like, I'm gonna make it to 100. But I think given like technological advancement and stuff, like we've got a good chance. So
do you think?
Yeah.
Really?
Sure. Yeah,
that's interesting that you think that. Okay, well, on that then, let's get into the science. So let's talk about what kind of technologies are actually on the horizon, or what work is actually being done, because I learned a thing or two from that book. So, but I can't really remember it. So let's get into it.
Yeah. So we are actually going to go into epigenetics. So I find it fun that you've already mentioned that. But there's actually so much research that's going into this. And I find it funny because there's a lot of famous tech people like Jeff Bezos and Sam Altman.
Yeah, because they're just narcissists, aren't they?
They just don't want to die. So they're actually funneling a lot of money into this kind of research.
So now they're like, now we have money to put into science all of a sudden.
Yeah, it's weird that, isn't it? So the first thing that I'm going to talk about is senescent cells, which I did actually mention earlier. So your cells are obviously not meant to last forever. They're not built to last forever. And so every time a cell divides, it has to duplicate its chromosomes. And due to how this process works, a small portion of DNA at the end gets lost every time. And that loss could be disastrous. So cells are protected by long stretches of DNA called telomeres. So like on the end of like your coded DNA are telomeres, which are long stretches of DNA, which basically are sacrificed every time a cell divides. And so they kind of function a bit like the plastic tip on your shoelaces. So they protect the rest of the shoelace. They protect it from fraying and like getting all up, basically. So like I said, these telomeres, they get shorter with every division, and eventually in some cells, after enough divisions, the telomeres are completely used up and the cell turns into something like a zombie, which is scientifically called a senescent cell. And these senescent cells, they stick around instead of dying, and as you age, they accumulate in your body and they damage nearby tissue and are associated with many age-related diseases such as diabetes and kidney failure. But the real question that scientists have, what if we could eliminate them? So we're going to eliminate like a cause of damage, cause of inflammation that's causing problems in the cells, and it is associated with aging. So researchers have actually genetically modified mice so that they could remove senescent cells whenever they wanted. And older mice that had these cells cleared out were much more energetic, their heart and kidney function improved, and they showed a lower risk of developing cancer. And in general, they lived up to 30% longer and were healthier compared to typical mice. But obviously, since genetically modifying every cell in the human body isn't really feasible, scientists need another method to target and remove senescent cells. But we've also got to think about how can this be done without affecting healthy cells. Now, normally, most cells undergo a programmed form of self-destruction when they're damaged, but senescent cells, they resist this process. So there is this drug called dasatinib, which is typically used in cancer treatment, and another compound called quercetin, which is a flavanol that's often taken as a dietary supplement and naturally found in fruit and vegetables. And researchers actually discovered that when these two were used together, they can selectively clear out senescent cells in mice while leaving the rest of the body's healthy cells largely unaffected. And basically, senescent cells don't produce a protein that causes programmed cell death. So it kind of forces the senescent cells to produce this protein and then essentially caused the cell death that needed to happen. And so the mice that this was tested on didn't just live slightly longer, but more importantly, they didn't spend extra time in the kind of frail, deteriorated state at the end of life. Instead, they maintained a healthier function for longer, and these mice actually behaved and appeared more youthful. So their fur was thicker with less graying, their skin appeared fuller, their eyes brighter, and they carried less of the weight gain, which is typically associated with aging. So overall, they had the appearance and vitality of younger mice compared to the control mice. And obviously, because of findings like these, there's several new companies that are now exploring therapies that target these senescent cells, and they're now termed senolytic drugs, and they're entering early human trials. So they may significantly improve health span, though like lifespan extension remains uncertain.
It's so interesting. So cool.
It is crazy to think of like the different aspects of aging and how like people tease these things out, and then how that then translates into animal models as well. And I think looking at mice is really interesting because they obviously have like a lifespan of about 2 years. So it's actually quite easy to study these things in mice. I'll go into it later, but it's obviously a lot harder to study these things in humans because they obviously live so long.
This episode is sponsored by Cite, the smart citation tool for your everyday research needs. Taz, I don't know about you, but I think Cite really sets itself apart from other citation and reference tools.
Yeah, I feel the same. With Cite, not only do you have access to over 250 million papers, but it can summarize a paper into a report, format citations, and more.
Yeah, and not only that, but I also love that you can connect connect it to tools like ChatGPT, Claude, or Copilot, and it basically just upgrades how those tools actually handle research.
Yeah, so instead of getting a general answer, your AI is actually searching through a huge database of research papers. And the key thing is it's not just looking at the titles or abstracts, it's searching inside the full text.
Honestly, it's such a slay because it just makes such a big difference, because a lot of important details don't really show up in abstracts, and I feel like most tools just miss that completely.
On top of that is the smart citations feature, which I think is one of the most useful features because it doesn't just show how many times a paper's been cited, it also shows how it's been cited.
Yeah, you can see whether other researchers have supported the findings, contradicted them, or whether they've just mentioned them. So it gives you a much clearer picture of what other experts in the field feel about it.
It also helps with access. So if a paper is available to you, Cite will help you get the full text.
So if you want to try it, you can head to cite.ai, or you can find the link in today's episode or video description. Setup only takes a minute, and you can use the code on screen now for money off. And thank you so much to Cite for sponsoring today's episode. But for now, let's get back into it.
So the next aspect is NAD+ and cellular metabolism. So basically cells are composed of hundreds of millions of moving parts that drive the chemical reactions keeping us alive, and this is often referred to as cell metabolism. So the components for this are constantly being broken down, cleared away and rebuilt, and as we get older, this maintenance system becomes less efficient. So cellular parts become damaged and they clump together, they're cleared more slowly, or they're produced in insufficient amounts. And one of these crucial components is NAD+, which stands for nicotinamide adenine dinucleotide, which is a coenzyme that plays a central role in helping cells maintain and repair themselves. So by the age of 50, levels of NAD+ in the body can drop to roughly half of what they were at the age of 20. So reduced NAD+ levels have also been associated with a wide range of conditions, including neurodegenerative diseases like Alzheimer's, cardiovascular disease, metabolic disorders, and certain cancers. NAD+ itself cannot easily cross cell membranes, so taking it directly as a supplement in that way isn't very effective. So instead, researchers have found that precursor molecules such as nicotinamide riboside, or NR for short, or nicotinamide mononucleotide, which is known as NMN, can enter cells and then be converted to NAD+ internally. So in 2016, several studies in mice demonstrated that boosting NAD+ through these precursors enhanced the activity of stem cells in tissues like skin, brain, and muscle. And the treated mice showed signs of rejuvenation, which included improved DNA repair, better mitochondrial function, and modest extensions in lifespan. So these findings even drew the attraction of organizations like NASA, which has explored NAD+ related approaches as a potential way to reduce DNA damage in astronauts exposed to cosmic radiation during long duration missions. And then since then, early stage human trials have been conducted on NAD+ precursors like NMN and NR, and these studies generally showed that they can safely increase NAD+ levels in humans, with kind of some evidence of improved metabolic health, things like insulin sensitivity and markers of cardiovascular system. But there's not really strong clinical evidence that they extend the human lifespan. As I said previously, it's kind of hard to measure this because humans live for so long, and the long-term effects of NAD+ supplementation is still under investigation. It's a very rapidly evolving field. Scientists are also studying its interaction with proteins called sirtuins. Sirtuins are basically a family of NAD+ dependent deacetylases and are often described as longevity genes, and they essentially require NAD+ to activate. So obviously supplementing with NAD+ activates these longevity genes, and that basically allows them to repair DNA, it controls metabolism, and it reduces cellular stress. So at the moment, NAD+ boosters remain promising, but experts kind of caution that they're not really proven to be anti-aging therapies yet, because it's kind of hard to prove that. So
Yeah, it's so interesting. Like, I hadn't even really thought about like how you prove that something is actually anti-aging. I mean, when I hear like
Mm.
anti-aging, I always think about like skincare sold to us as women.
Yeah.
Like 23 and you're like, oh, I need like this anti-aging cream. It's mental. But right, really thinking about like actual anti-aging, not just like how our skin looks or anything like that, is like,
Hmm.
yeah, it's really interesting.
I also think with like a lot of this work is kind of seeing associations with aging. So obviously with NAD plus work, they saw that someone at 50 years of age has half the amount of NAD plus as someone at 20. So then immediately they were like, that's probably associated with
Yes.
aging. And then they obviously look at the mechanisms behind it and find that they are like associated with like the longevity genes, because that is ultimately like what activates it and stuff like that. And I find part of me is a little bit like, but how do you know that just like supplementing with like this artificial NAD precursor is actually going to do what you want it to do?
Right. I mean, that is the whole aging field though, really. Like from what I know of the aging field, that is how it is. I mean, I'm sure you're going to talk about it later on in the episode, but it's the same with epigenetics. Is like, okay, we know as you age, you lose methylation. So then scientists are like, okay, let's see if we can restore or maintain that methylation better. But then like realistically, how do you know what that's actually going to do? Do you know what I mean?
Mm.
Because you don't actually know what the effect is of losing that methylation realistically. Do you know what I mean? Like, yeah, so I think that is
Mhm.
the majority of the aging field is like, we can see that this happens as you age, but we don't really know what that means. But let's just see. It's very interesting. It's definitely one of those fields where I feel like people have been working in the field for ages and they still don't really know what's going on, which I think makes it an even more interesting field. But when you were talking about NAD plus, I feel like I've definitely heard of it before. And do you know how I've heard of it before? It's because I
Mm.
feel like I've seen, and you might be like, no, Liv, I don't know what you talk about, but I feel like I've seen celebrities like raving about like NAD plus infusions.
Yeah. Yeah. When seen Hailey Bieber talk about NAD plus infusions, and I was kind of like, I'm sorry, like just another thing for us to have to
I
consume.
know,
Like,
don't,
and then
yes,
I
I
was actually
know.
quite surprised to see that it's kind of rooted in some actual science.
Interesting. We'll be lining up to get our NAD plus infusions. No, I'm joking. As women, listen, there's not another thing we need to consume, and that's a whole nother topic for another day. But do you know anything about stem cells? Because I also feel like stem cells is a big theme in
Yeah.
this topic.
Yeah,
So,
it is a big part of it, because basically, if you don't know anything about stem cells, they are kind of like your body's templates. So it's
mhm.
basically a group of unspecialized cells that reside in different tissues, and they can continually divide to generate a steady supply of new and healthy cells, and they can basically turn into any cell that is required. That is the whole point of a stem cell, is that they haven't differentiated in a specific cell type. But over time, however, their numbers and effectiveness declines. And as that happens, the body's ability to repair and maintain itself also declines. So in mice, scientists observed that as stem cells in the brain diminished, the animals became more prone to disease. And to test this, researchers actually extracted stem cells from the brains of young mice and then injected them into the brains of middle-aged mice, specifically into the hypothalamus, which is a small but critical structure involved in regulating many essential bodily functions. And these newly introduced stem cells appeared to rejuvenate older brain cells by releasing microRNAs, which helped regulate cellular metabolism. And after several months, the treated mice showed improved brain and muscle function compared to the untreated mice. And on average, they lived about 10% longer. So basically, your stem cells kind of act as like this little storage facility where like they can be taken out of if you need repair anywhere. It could be in your brain. A really good example is like when you break a bone, stem cells will kind of like fill the gap and repair your broken bone, essentially. In another experiment, scientists took stem cells derived from mouse embryos and injected them into the hearts of older mice, and the results were really striking. So the mice demonstrated better heart performance and were able to exercise for around 20% longer. And most recent research has expanded on these findings. So scientists are now investigating not just transplanting stem cells, but also activating the body's existing stem cells or using stem cell derived signals like exosomes or growth factors to trigger repair. So early human clinical trials, particularly using mesenchymal stem cells, which are basically stem cells that are found in like your bone marrow and things. I feel like a lot of people have heard of like stem cell therapies. It's kind of linked to that, but kind of relating it to aging. Some trials show modest improvements in tissue repair and inflammation reduction, but results are still kind of mixed. And obviously there's a lot of implications with safety and ethical considerations, especially when it comes to embryonic stem cells as well. So stem cell therapies, they might restore tissue function in humans, but ethical and regulatory challenges still very much remain.
Yeah, it's so interesting. I just think stem cells in general are such an interesting area of science. But obviously, I don't know if any of the listeners know, and maybe you'll tell me that I'm wrong, but I feel like getting access to stem cells is quite hard and it's like quite a limited thing.
Yeah, it is. Mesenchymal stem cells, like I said, they're typically found in bone marrow. So it can be obviously quite invasive to access, like humans' own stem cells, and kind of seen as a bit like pointless just for aging alone. But I actually found this next aspect of like anti-aging research really interesting. So similar to the effects of stem cells, we also have an area of research called cellular reprogramming using the Yamanaka factors.
It feels like such a throwback to me. This is how I got really interested in like epigenetics, actually, because I bought the aging book and then I bought an epigenetics book, and in the epigenetics book they talked about the Yamanaka factors, which I'm assuming you're going to talk about in a minute, but it's the transcription factors, right? Yeah, so it's like there's some transcription factors, and Taz will tell you about it in a minute, but then my dissertation project, like my final year project, I did on one of the Yamanaka factors, so I just feel like
that is interesting. I didn't know that.
Yeah,
that is cool. Well, you're like the epigenetics part even more then.
I'm excited.
Okay, so cellular reprogramming is basically based on the idea that babies born to parents in their 30s essentially have the same life expectancy as someone born to parents in their 20s or even in their teens. So despite being created from older cells, so an egg from a 30-year-old female and sperm from a 33-year-old man, for example, the resulting child begins life with a reset biological age and a normal life expectancy. So a similar life expectancy to someone who was born from a 20-year-old egg and a 22-year-old sperm, for example. So in a sense, this suggests that biology already has a built-in way of resetting the aging clock. The real question is whether we can understand that process well enough to replicate it deliberately. So in laboratory settings, scientists have started to do exactly that through a technique known as cellular reprogramming. So Shinya Yamanaka's discovery of 4 key genes, now known as the Yamanaka factors, and their introduction into an adult cell can reprogram the cell. And this approach was originally developed for stem cell research, where the goal was to take an ordinary adult cell, so a skin cell, for example, and rewind it back to a pluripotent stem cell, which is equivalent to a stem cell that hasn't undergone differentiation. And then it is then capable of becoming almost any cell type in the body. So what researchers discovered along the way was particularly intriguing. As they reversed the developmental state of these cells, they also appeared to reverse aspects of cellular aging. So in other words, the cells didn't just become more flexible in what they could turn into, they also became biologically younger. And building on this, scientists began testing partial reprogramming in whole animals. So in mice, they found that instead of fully resetting cells back to stem cells, which can actually lead to issues like tumor formation, they could dial the process back. So this partial reprogramming didn't erase the cell's identity, but it did seem to show reversal of age-related organ decline. So it restored vision and improved muscle function. And because of this potential, this area has attracted enormous scientific and commercial interest. So researchers are now exploring whether controlled reprogramming could one day be used to rejuvenate tissues, treat age-related diseases, or even extend human health span. Today, current efforts by companies like Altus Labs and Retro Biosciences, which, like I said, are funded by Jeff Bezos and Sam Altman, are trying to translate this into clinical therapies.
Listen, if they've got Jeff Bezos and Sam Altman on the case, probably not that unrealistic. Money can buy you anything, and they are rich. So if they're interested in it, it will probably happen. Yeah, it's also interesting, although I can't help but just be excited to get to the epigenetics. Not that I'm rushing you ahead, but I feel like it's just one of the most interesting parts. So
it is pretty interesting, and obviously something you're going to be fully aware of. So if at any point you're like, I'm not sure about this, Chaz, just buy it because you're the expert on this. So the first thing we're going to be talking about is epigenetic clocks. So to estimate your biological age, you might have to provide a blood or a saliva sample, and scientists can analyze specific markers on your DNA. And these markers, often chemical modifications like DNA methylation, they can change in predictable ways over time, and they appear to be closely linked to lifespan and overall health. So when researchers first developed looking at these epigenetic clocks, the goal was fairly simple. Could you accurately predict someone's age based purely on these molecular changes? So imagine I was to take a epigenetic age test. I'm 30, but say the results suggest my biological age is 40. What scientists found is that people with this kind of accelerated epigenetic aging tend to then have a higher risk of disease and an increased likelihood of earlier death compared to those whose epigenetic age matches or is even younger than their chronological age. And that's what makes these clocks so powerful. For the first time, they give researchers a way to accurately measure biological aging rather just infer it from someone's like chronological age. So they're now being used to study how lifestyle, environment, and potential therapies influence aging at a cellular level. But here's where it starts to connect with genetics as well, and where things get really interesting, because epigenetic changes don't happen in isolation. They're deeply tied to the same genetic pathways that control how our bodies respond to stress, damage, and even starvation. So the idea that aging could be altered through these pathways actually goes back nearly a century. So in the 1930s, scientist 𝗾𝘂𝗲𝗯 showed that restricting calorie intake in rats without causing malnutrition could extend their lifespan by up to 33%. And this phenomenon, known as dietary restriction, has since been observed across a huge range of species, from yeast all the way to mammals. And importantly, it doesn't just extend life, it also extends health span as well. And from an evolutionary perspective, so we're going back to that, it actually makes sense, because in times of scarcity, organisms shift resources away from growth and reproduction and instead invest in survival so that you can do reproduction when you're not just trying to survive. So it enhances things like repair system, it improves stress resistance, and it maintains the body until conditions improve and until you can reproduce, basically, because that's what it's all about, reproduction. And this is where genetics come in. So in the 1990s, scientists studying roundworms discovered that altering a single gene called DAFT could double their lifespan. And these worms didn't just live longer, they stayed healthier for longer, which showed an increased resistance to stress, better DNA repair, and improved immune function. And what's remarkable is that this gene is part of a broader evolutionary conserved pathway, meaning similar systems exist in humans. So now we kind of have two pieces of the puzzle. On one hand, you have the genetic pathways that control longevity, and on the other hand, you have epigenetic clocks that measure how fast aging is actually happening. So rather than changing DNA itself, so like introducing genetic mutations to get an outcome that you want, that would be a permanent change and difficult to control. Scientists are increasingly focusing on epigenetics, or the epigenome, which is the layer of regulation that controls how those genes are turned on and off. It's been widely studied that environmental factors like diet, stress, and lifestyle feed into this system and influence which genetic pathways are turned on or turned off. So calorie restriction, for example, appears to slow aging not by rewriting your DNA, but by shifting epigenetic signals, essentially telling your body to prioritize maintenance and repair. And that shift is something we can now see reflected in epigenetic clocks, which is why this field is so promising, because if aging is at least in part driven by changes in gene expression, then in theory it might be reversible. By resetting epigenetic markers, scientists have already shown in animal models that cells can regain more youthful patterns of gene expression. Biological markers of aging can be partially reversed, and in some cases aspects of aging can be slowed or even turned back. However, whilst this is incredibly exciting, it is obviously still in very early days. Epigenetic clocks are really powerful research tools, but they're not quite ready for like widespread consuming use, because we don't fully understand what drives all the changes, like you said before, Liv, or how we can reliably intervene in humans. So if you took one of these tests today and it told you you were biologically older than expected, the big question remains, what do you actually do with that information? Right now, the answer isn't entirely clear. These clocks are highly informative, but not actionable, because obviously epigenetics can link to things like starvation or healthy eating and stress. So basically, the main advice right now is to live a healthy lifestyle, exercise, because epigenetically, they've seen that that is related to a lower biological age. So yeah. I hope I did that justice, Liv.
No, that was a good like general overview. I mean, obviously, I've been deep in the field for like a few years, and I actually just have something to add to it, which is there's like clocks that have been made for everything. So we've got like an aging clock, but there like are actually so many clocks for things, and I use them all the time in my research. It's not really a clock, but it's very similar to how a clock works, but we have a tool that can predict like if you are a smoker, if you've ever smoked, if you've never smoked. You have ones that can predict like sex. Like I work a lot in the field of like epigenetics and just like sex. So I work a lot on like sex differences and stuff. But I was part of a paper where we like made a tool that you can look at people's epigenetics to predict their sex. So there's loads of different ones, but I find it interesting, like what you said about if they were to be made commercially available, then yeah, absolutely. What do you do with that information? Okay, your biological age is younger, but like, what does that mean? What do you do with that information? So I feel like now they're just more used as like a really good tool for researchers. So we would use it a lot as like quality controlling on our data. So like if they've been reported to, like this would happen all the time. So this is like such a big important part in like quality checking my data, is that I would have people who would report themselves as like a male, and then we'd look at their like epigenetics and they would appear as like a female. Or we'd see a lot of like sex chromosome aneuploidies. So like someone would report as a male, but they'd actually like epigenetically appear as like XXY. And it happens all the time. So like for us as researchers, like that's an important tool, because most times we'll probably remove those people like from our research, because you know things aren't adding up in terms of like reported and then like what we're seeing biologically. But it's all very, very interesting.
That's so interesting. Yeah.
But anyway, my point to this was that the guy who you're talking a lot about like aging clocks in epigenetics, and the main kind of researcher who worked on it was a guy called 𝗾𝘂𝗶𝗯, and he's like really famous in this field. But he actually also made this thing called 𝗾_age, which is essentially a clock that you can like it's the same thing as the aging clock basically, but it apparently tells you how like when you're going to die. That's like a very
Really?
like general view on it, but yeah, it's basically the whole idea behind it. It's like it's gonna tell you like when you're gonna die based of your methylation, and like yeah, it's really interesting, and you should go and read about it. But they've made these clocks not just for humans, but for like tons of different animals. Like there's so many different animals with these clocks as well, so it also works on them. So it's very, very interesting. Anyway, I'm gonna shut up because I could talk about it all day because I find it very interesting. But
If you had
yeah.
the option of like doing that grim age thing, would you do it?
Well, yeah, because I just don't think like epigenetics is the only thing that's predicting when I'm gonna die. I think it's like a very big part of it, but I think it's way more nuanced than that. Do you know what I mean?
Well, yeah, I was gonna say I wouldn't do the grim age thing. I'm happy not knowing that, I think.
It's probably just a similar, you know, I never looked into properly like how it worked, and I've actually never read the paper. So I probably should, and maybe one day we can do an episode on it if people are interested. I imagine it probably is some kind of similar information to just the general aging clock, but yeah. Again, what do you do with that? It's exactly what you said. What do you do with that information?
So like, I don't obviously want to boil it down to, I don't know, just a questionnaire. But if you were to have a questionnaire among like loads of different people, and well, you do when you go to the doctors and stuff, and like they ask you things like, do you smoke? How often do you exercise? Do you eat a balanced diet? Obviously, these are all things that we know extend your lifespan or your health span or whatever associated with longevity. I just feel like epigenetic aging clock is just like a scientifically advanced version of that, because it's essentially showing like
That's so interesting to say
these
that,
things
actually,
and how
because
they link to
yeah, it's
your
very interesting to say that,
discussion.
because I actually think like these clocks, if they become like commercially available, could be like really dangerous, right? Because obviously you have the ability now to go to the doctor and lie. Like if the doctor says to you, do you smoke? You can say no. Say you're an American and your insurance provider asks you, do you smoke, right? Let's think about the smoking epigenetic clock. So that clock, some of them can actually predict secondhand smoking. So me, for example, I grew up with parents who smoked a lot. Like my grandparents smoked a lot to the point where like their ceiling is yellow. My dad smoked, my mom occasionally smoked. So like I was inhaling a lot of secondhand smoke, which obviously like back then, you know, we wasn't that bothered about. Now we are more bothered about it. But I wonder if, you know, that would show up on my epigenetic clock. So it's like if you make that commercially available, and then now insurers have like insurance companies where eventually one day to get access to that, and then they can raise my insurance premium because they're like, you've been affected by secondhand smoke. So I mean, so I think that like this whole thing could be quite like problematic. You could make one for like alcohol consumption, and like, not that I think we should be lying to our insurance providers, but also like that is a whole conversation in itself, like the socioeconomic impacts of that. But I just think it is a very interesting topic. It is, but I do also think like when you're talking about people like Sam Altman and Jeff Bezos like getting involved in, I'm like, oh, I don't know how. So yeah, I mean, we could totally do an episode on this because I could talk about this for a long time. So if the listeners are interested in learning more about this, then definitely let us know. But yeah, it's a very interesting topic, and yeah, but I am curious. Are there any drugs in the aging field that are being used? We've mentioned like supplements and things, but are there any actual drugs?
Yeah, so there's definitely drugs that show some promise in slowing aspects of aging. And I basically want to point out that aging is basically a result of chronic inflammation, which then leads to cell death and damage and reactivation of the immune system, where the cycle essentially repeats. And like as we age, it gets more and more, and there's more cell death and like stuff like that. So that is essentially what we're trying to prevent here. So drugs targeting inflammation and how our immune system responds have shown the most promise. So as I mentioned earlier in the podcast, there's a class of drugs known as senolytics, which target senescent cells, and they're being tested, and there's numerous clinical trials underway to test all those different therapies in humans. Other drugs already in use for actually different conditions are also being investigated for their anti-aging potential. So you may have heard of metformin, which is a widely prescribed treatment for type 2 diabetes. It appears to reduce inflammation and improve cellular metabolism. So observational studies suggest that people taking metformin may live longer and have lower rates of age-related diseases, even compared to non-diabetics. And rapamycin, which was originally developed as an immunosuppressant for organ transplant patients and have actually shown anti-aging therapy in animal models. And it basically works by inhibiting mTOR, which is a protein that drives aging and age-related diseases when it's overreactive. And this basically improves cellular function, and it alters how cells sense and process nutrients, and it promotes the breakdown and recycling of cellular waste. And what I find really interesting about rapamycin is they've actually started prescribing it. I don't know if it's all globally. It might just be in the US. It might also be in the UK, but I'm not sure. But I find it really interesting. You can now buy it as a drug to give your like elderly dog. And there's been like anecdotal reports of people like giving it to an elderly dog with like joint pain and like chronic inflammation and like chronic conditions associated with aging. And there's like anecdotal reports that after they give them this drug, they are rejuvenated and they don't have like this chronic inflammation and their joints aren't
Wendy.
like as up. So yeah, I find it really interesting that that's
It's interesting.
a thing. But obviously, despite all of these advances, there's actually no drug currently approved specifically to target aging in humans.
Yeah.
And one of the main challenges is the time required to demonstrate effectiveness in humans. Obviously, as we've mentioned, because proving that a therapy extends both lifespan and health span could obviously take decades because we live so long. You do obviously have things like the detection of new biomarkers that are associated with biological age, like we mentioned with epigenetics. So like obviously if you give these therapies that are meant to promote anti-aging and then follow through and have these epigenetic changes that are associated with younger age, then it might help accelerate the process of determining whether it is actually anti-aging in humans. But obviously it's a nuanced process. It's quite a long process.
No, 100%. And like going back to epigenetics, like you just said, like lifestyle is obviously like a really big factor in anti-aging, as we've kind of spoke about throughout this episode. But I almost feel like sometimes maybe we're just like uncovering the mechanisms behind what it means to live a healthy lifestyle. And like that's obviously related to aging, you know?
Yeah, I 100% agree with you. And when I was doing the research for this, I think, yeah, what you just said, I think we're kind of just uncovering what it means to live a healthy lifestyle. Like there's a reason doctors tell you to eat a healthy diet and exercise, and it's because those things are associated with living a longer life. And a lot of the time we probably don't know the extent to which things actually promote healthy aging. We just know some of the reasons. Like obviously if you eat a diet low in fat, then you're less likely to have heart disease and things like that. But I think at a molecular level in cells, we're only just starting to like really uncover the extent of those reasons. And I think the more we learn about anti-aging therapies and the pathways effect, we typically see these things linked to a healthy lifestyle, especially epigenetically as well. And I think also in my research, I get so interested how like smoking is associated with microbiome changes in the gut and how that then leads to colonization of the gut with bacteria that also promote inflammation in the rest of the body. And so not only is your diet then going to prevent the colonization of bad bacteria if you're eating a healthy and diverse diet, but also if you don't smoke, then that's not going to happen in the first place. So I find it so interesting that actually it all does actually come down to what it means to live a healthy lifestyle. And
it all comes down to like basic shit, right? Because don't you think sometimes in science we're trying to do the absolute most, like, oh my god, how do I like hack my biology to, and it's like, babe, just, you know, eat well, sleep your 8 hours, like exercise. Like, these things are actually what you need to be doing.
Yeah, 100%. I think obviously for people who have lived their life and, I don't know, not eating healthily, it would be great to like have a drug where you can give them it and be like, hey, despite what you've done and you've not eaten healthy or you've smoked or you've not been able to exercise as regularly, here's this drug that is just gonna prevent those bad things associated with that.
Yeah, no, I agree. But anyway, that's again a whole nother conversation that we could have. But a big philosophical question to end on. Even if we say we figure out how to stop aging, like completely, should we?
Yeah. I think going into this, like doing the research for this, I was very much like, no, you know, like we should all like grow old and die because that is the nature of things. And like, to an extent, I still believe that. But I think extension of health span is like super important because nearly half of healthcare spending occurs in senior years, and much of it manages age-related diseases. So if you could like not necessarily extend lifespan, but if you could halt aging, it could reduce that burden, and it would give people more time to learn, work. Maybe that's not such a good thing,
No,
but
literally,
like, I
that's
don't
exactly
know,
what I was
just
thinking.
live.
Fuck, they'll make us work longer. Like, retirement is going to come even later.
Yeah, that is probably true, to be fair. Like, if we are healthier for longer. But I don't know. I think if we've got the option to live a long life that is kind of not associated with like an aching back and painful knees and I don't know, all the things that you associated with getting older, then I think it's probably a good thing.
Yeah. No, I agree. I think like expanding health span should be the ultimate goal really of this. But can you sum it up for us? What do you think we can expect in the future when it comes to this field?
Well, essentially aging is no longer this like untouchable process. Like, it's something that we are working towards to halt or slow down, because essentially it's just a complex network of biological changes, which is usually influenced by molecular damage, cellular decline, stem cell exhaustion, and epigenetic drift. So the science of aging is moving from theoretical biology to actionable interventions. So within lifetimes of people alive today, we may see therapies that remove senescent cells, rejuvenate stem cell populations, reset these epigenetic changes, reprogram tissues for partial rejuvenation, and then boost cellular repair with targeted drugs. And those are all sorts of things that would prevent the aging that we see. But obviously, it's not associated with one single solution. My question is, if you could choose today how long to live, I guess we've kind of already touched on this, in health and vitality, what would your answer be? Because I actually read somewhere that if you are 50 now, you can expect to live up to 120 years old. So with science accelerating at this pace, the future where we're like living until, I don't know, 150 may not be impossible.
Oh, I hope my mum lives until 120. That'd be great.
Yeah, me too.
Unfortunately, I can't get a letter from the Queen anymore. Do I want a letter from the King? Nah, I want to be a centenarian, a living legend.
Yeah, I agree. But then it might just become normal.
Okay, well, I want to be a
Everyone lives
200 girly then.
200. I mean,
we're a centenarian and a half, okay? That's what I want to be, a centenarian and a half. Guys, let us know in the comments on today's episode, what age would you want to live to? Because I'm intrigued. 100 centenarian and a half.
Maybe by that point we'll be like, God, it's going on for too long.
I reckon so, you know, that's really sad, but I reckon so. Babe, our pension will run out. That's what I'm saying, they're going to make you work longer. We'll be working until we're a centenarian.
Oh my God, imagine me being on the bench handling viruses at 100 years old.
100, that's iconic.
Probably not
that is
smart.
So iconic.
Probably not smart. I'd probably like create a pandemic or something.
Anyway, this episode was so interesting, Taz. Thank you so much. I really, really enjoyed it. Guys, let us know what age you want to live to. But for now, I think it's about time we get into the laboratory section of today's episode, and we have a very juicy story, so let's get into it. Okay, so this story is a bit crazy, and it starts off by saying, I think I'm being sabotaged in my lab.
Oh no. I'm
So
scared.
let's get into it. So, a bit of a long backstory. In my first year of my degree, I did a work placement at a lab my uni is affiliated with in the summer. There was a PhD student who was working there. Let's call her Beverly. Beverly was always really cold, standoffish, and not chatty in the slightest. Can I just say, I hate people like that. Sorry. Can you just be friendlier,
Can't
please?
relate.
I like I like a friendly person. Anyway, so she's not chatty in the slightest, but fair enough with the PhD stress. I get I must have just been an insignificant undergrad to her. Fine, I didn't think anything of it. Second year of my degree, she was helping out in the labs at the uni, and we were doing an experiment where our results were required for an assignment. The experiment was a bit of a shit show, first of all, because the original lecturer left. Second of all, because it was a large class and we all had to work in one fume hood. I don't really get that. What does that mean? Like, that's bad?
They're just sharing like a piece of equipment, essentially, which is...
How annoying is that on a scale of 1 to 10? The listener wrote it was long as... Anyway, she says, I accidentally missed a step in the protocol, and then Beverly afterwards informed me that she saw me miss something. I did passively aggressively say, thanks for telling me, lol. So I had to start completely from scratch with everyone else waiting to use the fume hood. That would be me. That would be me.
Same,
I am that bit.
Why are you telling me now when you could have told me when you watched me do it wrong?
I can't lie, like, it is already giving like sabotage, because like, why would you not just say something like, surely you guys are working together as well? Like, if your lab partner misses something, you'd be like, what are you doing? Like, do you tell them in the moment?
Yeah, I agree.
So it's already seeming a bit odd, I'm not gonna lie. And then like she said, she's got to wait for everyone else to use that fume hood, baby. She's waiting even longer. Then she goes on to say, I am all for learning from mistakes and don't easily get embarrassed, so that wasn't why I was annoyed, but we literally didn't have time for that bullshit. Third year, I'm doing my final year project at the same lab I did my work placement at, and Beverly has recently started working as a lab tech there. Oh, that would annoy me so much. She goes on to say, which I'm annoyed about anyway, but the old lab tech was an absolute babe, so it's even more annoying. I love this listener.
Same.
My supervisor and Beverly are really chummy. Of course they are. And my supervisor assumed we hadn't met, so introduced us. And Beverly immediately was like, oh yeah, I know her. This is where it all started to feel personal. Beverly has been super friendly to me since I've been doing my project in the lab, now with my supervisor, but it seems so extra and fake as. And then a couple of times when I had been alone with her, and she has been much sassier with me, and I always feel like she's accusing me of something. For example, she asked typical health and safety questions, making sure that I'm following guidelines, which I always do, but it almost feels like she is trying to find something to blame on me. Also, since she has started, I have had a couple of contaminated cell culture flasks. Not saying it's her, but I wouldn't be surprised, lol. I'm not afraid of... Sorry, I love this listener because they finished it with, I am not afraid of a little bit of confrontation. So should I just ask her what the fuck her problem is? What would you guys do? this listener is sassy. Like, you're saying Beverly's sassy, babe. You are sassy. I'm going to take it back to when they were doing the experiment and she saw her miss the step, okay? I can't lie, that is definitely a red flag, because why would you not just be like, oh, babe, like, you've missed this, or like, oh, like, we didn't do this step, like, let's take it back. Like, the fact that she like waited until the end when the listener was like, had obviously queried something, and then she was like, it was giving, like, I could just picture it, like, she was like, well, yeah, you missed that step, like, really, like, do you know what I mean?
Yeah, I don't really know why she would have, like, if she saw it happen and then said it after, it doesn't really make that much sense.
Exactly, because it's just giving you weren't doing your job properly, because your job is literally there to supervise people and help them. And if you don't want to do that, then don't do the job. I'm sorry, but I don't think that's really fair. Like, as undergrads, you're going to make mistakes, you're going to miss steps. Like, the reason there's other people in the room is to teach you and help you. So if you don't want to do that, then don't do the job. Yeah, I think that was out of order, but not that deep, but like out of order. But anyway, so she goes... She's doing her PhD now in this new lab. Beverly's there. Beverly's being really nice to her. This is a good It's a good sign. Maybe Beverly was having a bad day that day because she says that Beverly's being really nice to her now.
Yeah, but she says it's sassy and like she's trying to insinuate she's done things wrong
Yeah.
by asking her sassy questions. Like,
I'm kind of
did you
getting the
think
vibe that Beverly's just a bit of a wannabe know-it-all and a bit of a teacher's pet. That's the vibe that I'm getting. Do I think she has a personal vendetta against you? Maybe, but it seems like she's just a bit of a teacher's pet and likes to be a know-it-all.
Look, I reckon she's just been awarded her PhD, right? She feels like she knows her especially over someone who's just come into the lab after our undergrad. So I know of many PhD students that like finish their PhD and then they're like, including
They
me,
think
by
they're
the
hot
way, like
shit.
I'm not
Yeah, they think
excluding
they're hot
me
shit.
from this. They think they are hot as shit. And then
It's
you
a canon
soon
event.
realize
Like, you're like, I've just become a doctor. Like, I am hot shit. Like, don't try and tell me I'm not. That's okay. You can have your moment.
Like Beverly in this case is hot shit, but like she's making the mistake of then being like turning her nose up at people below her, which I 100% don't do or don't condone because look, we're all just trying to learn out here. It probably comes across patronizing.
I agree. Like we were all that annoying, like undergrad. We were all that annoying first year PhD student. And like me personally, I had such nice like third year PhD students who put up with my bullshit. So like I'm always gonna be nice to them because I'm just like, oh, you know, like they just want to learn. They're enthusiastic. Like I get it. They can be annoying sometimes, but like as long as you set boundaries and you maintain boundaries, like there's no reason that you should have to be patronizing or rude towards them. So I don't with that. Like I think it's rude and it's like a power trippy. I don't like it. Like be nice to younger.
I agree. I think like that obviously everyone feels like they're that when they finish their PhD, but like some people
Which you are, by the way,
are on another level entitled and like think they're the and it's just like calm down, mate.
The next thing, right, is so I think we're thinking that Beverly's just on a bit of a power trip. At the moment, I'm feeling like it's nothing personal towards you. I think maybe this is just like her personality. But the part at the end, she says, since she started, she's had a couple of contaminated cell culture flasks. Not saying it's her, but I wouldn't be surprised. Okay, so this could be like one of three things, right? It could be that Beverly's absolutely not contaminating the cell cultures and like something else is happening. Or it could be that she is, but it's not intentional. Maybe she's just not that great at her job. Or she is doing it and it is intentional.
Or it's the listener and they're not very good at not keeping their cells free from contamination.
It could be any one of those three things. And how are we to know?
I do think that it would be so unhinged for someone to purposely contaminate someone's cell cultures.
Right, but you know, people do this. We've had these stories before. People do this. They do.
But it's more likely that you just
people
like
have banged on the lab benches. I do not put anyone past doing a little bit of contamination of the cell cultures. All right, people are out here having sex on lab benches.
Yeah, but I just think you're more likely to have like just touched your pipette tip on like a surface and then accidentally pipetted with it. Like,
or Beverly could have accidentally done it as well. Anybody could have accidentally done it. If there's multiple people working on it, any one of you, and even if there is multiple of you, like who have access to these, even higher chance surely of them being contaminated, right?
Yeah, if you're using the same cell cultures, then yeah, it could be anyone who's using them. For sure.
So in terms of like whether you should confront her, I personally would save yourself the embarrassment and don't confront her in the sense that don't be like accuse her of anything. If it was me, the next time she was to ask me like one of these, because it's giving like I feel like you are feeling a little bit patronized. So next time she asked me one of these like patronizing questions, I would just give like a very firm response back. And just, you know, like what's the word I'm looking for? Like establish. This is not like the best wording, but like establish your place and just like set those boundaries. But I wouldn't like confront her of anything because I guess like right now you don't have any evidence that she's actually done anything. So you've either got to be really sneaky and like set a trap and catch her if you really think she's doing it. And then I guess you could confront her, or you just have to like ignore it and just shut it down.
I almost feel like, you need to say something muggy back. Like,
yeah. Not that we're like encouraging you to be paid. But
no, but like if she
even
says something like blatantly obvious, like, I don't know, did you make sure that you shut the -80 freezer? You'd be
nah, I
like,
wanted to leave it open all
nah,
night,
left
babe.
it wide open and everything's defrosted. You know?
Yeah, 100%. Like, I can't lie, that's what I would do. Because sometimes people do just ask me questions where you just think, do you actually think I'm that thick? Like, it genuinely, it is insulting sometimes. Like, I
Yeah.
feel like I've definitely had like colleagues who have said stuff to me, and I just think, obviously I've done that. Like, what are you talking about? Like, obviously, like, how stupid, like, you have no reason to think that I am that stupid to not do that. So I get it is really annoying. So I agree. You've just got to say something muggy back. Because I think at the moment, maybe you're just like, yeah, like I did do that. And like, she's just enjoying this like power trip of feeling like she's telling you what to do. Or maybe she's just like genuinely
Also,
very
you can brush that off as a joke as well. You can be like, nah, left it open. Everything's defrosted now. All your samples are gone. And you can just laugh it off. But like kind of sets a precedent of like, obviously, like, I know what I'm
Yeah,
doing.
100%.
But
But
then I
also
do
some
think
people,
like she is more like experienced than you. Maybe she feels
and
like
also
she's
she's a lab
giving
tech,
you
right? So it
advice.
is her job. Like
Yeah.
maybe she's
Oh,
thinking like, I want to do my
no.
job properly now. The job of a lab tech is to like maintain the lab, help people out on like certain things. I think like your options are sassy remarks back, but in a friendly, laughy way, because you don't want to cause any tension. You don't want to have confrontation in the lab at work. Like ultimately, that's not what you want, and we don't encourage that on this podcast.
No.
Ultimately, I think she's just probably a bit of a know-it-all and is
On a
just
bit of a
trying to do her
power
job.
trip.
Yeah, just trying to do her job, like being a bit too extra. You know, like when you go to Tesco's and you get ID'd for a Red Bull and it's like, babe, like
Yeah.
chill out a
Come on,
bit.
Karen.
Stop being a ride or die for Tesco. Like just let me have the Red Bull. Because I genuinely sometimes I've been denied like they won't sell me the Red
Yeah,
Bull
so
because
have
I don't have an
I.
ID.
So have
And I'm
I.
like, oh, for God's sake, like stop being a ride or die for Tesco. Like it's not that deep.
Yeah.
Just let me have the Red Bull. I get it. You're not going to sell me
Or
a
like
bottle of
super
Tesco,
glue
but
or
the Red Bull,
something.
huh?
Super
Super
glue or
glue. I'm
something.
like, for God's sake. I get you not selling me the bottle of vodka. That's fine. I can get on board with that. But like, let me have the Red Bull and the super glue, you know? But
Yeah.
some people are just like that, you know? Maybe she's changed since her days.
Also, it's her first job out of her PhD, probably. She's probably like, oh, I want to do everything right. And it's probably coming off condescending and annoying, to be honest.
But it could be worse, right? Like you could have someone who genuinely is like a proper arsehole and they are like genuinely like just really making your life hell. So if someone's being a bit of a know-it-all, it could be worse. As far as I'm concerned, especially when we're in academia, things could be a lot worse. So
yeah,
you know, make your little sassy remarks. Hopefully she'll stop. And also it's kind of part and parcel of being a younger PhD student. Unfortunately, sometimes you do just have to deal with people like this. But listen, the longer you're there, the easier it's going to get and probably the nicer she is going to be. So make your sassy comment back and forget about it, I think. You got bigger fish to fry, experiments to do, thesis to write, babes. You got this. You know what I mean?
I agree.
Anyway, with that rounds off episode 3 of What If. Make sure you guys tune in same time next week for a brand new episode where we are going to be talking about, it's a surprise. You're going to tune in next week to find out. So we'll see you in next week's episode. Bye everyone.
Bye.